Assalamualaikum and hello everyone. Today we will present our topic about apparatus, technique and reagent in analytical chemistry. Our application focus on the determination of acetic acid concentration in vinegar using acid-base titration. Through this presentation, we will discuss the laboratory apparatus, analytical technique and reagent involved in this analysis.
First, let me introduce what is analytical chemistry. Analytical chemistry is the branch of chemistry that focuses on identifying and measuring chemical substance. Accurate analytical results are important in industries such as food production, pharmaceutical, and environmental monitoring. To obtain reliable results, appropriate apparatus, correct techniques, and suitable reagents.
Our selected application is the determination of acetic acid concentration in vinegar using acid-base titration. Vinegar contains acetic acid as its main component. By reacting vinegar with a standard sodium hydroxide solution, the concentration of the acetic acid can be accurately determined. This method is widely used in food quality control to ensure product consistency and safety. There are several basic laboratory apparatus are commonly used during analytical procedures. First, beakers are used for holding and mixing solutions, while conical flasks are used during titration because their shape allows easy swirling without spilling. Another is measuring cylinder are used for measuring approximate volumes, and wash bottles are used to rinse glassware with distilled water. These apparatus support the analytical process efficiently.
Next, analytical balance are used to measure mass accurately. There are two common types, which is top loading balance and analytical balance. Analytical balance provide higher precision and are suitable for preparing standard solution. Proper care includes keeping the balance clean, avoiding overloading, and ensuring the balance is placed on a stable surface to obtain accurate measurements.
Hi everyone, my name is Nur Amana Batrisha and I will be presenting on volumetric glassware. This slide shows the main types of volumetric glassware used in volumetric analysis and acid-base titration. Volumetric glassware refers to laboratory glassware designed to measure and transfer liquid accurately. The first type is the pipette, which is used to transfer an exact volume of solution from one container to another, such as transferring a vinegar sample into conical flask. The second type is the burette, which accurately delivers variable volumes of titrant during titration. For example, sodium hydroxide solution. The third type is the volumetric flask, which is used to prepare solutions to a fixed and exact volume, especially standard solutions. These glassware are important because they ensure accurate measurements and help procedure reliable and accurate titration results. Next, I will explain the calibration of volumetric glassware. Calibration is the process of checking the accuracy of volumetric glassware to ensure the actual volume matches the state volume. First, the glassware is filled with distilled water. Then, the mass of the water is measured. Next, the volume is calculated based on the mass of the water. Finally, the calculated volume is compared with the state volume of the glassware. Calibration is important because it improves the accuracy of measurements, reduces errors, and ensures reliable experimental results. That's all.
Next, I will explain the reagent used in the experiment, which is sodium hydroxide or NaOH. NaOH is a strong base that was used to neutralize acetic present in the sample. During the titration process, NaOH reacts with acetic acid to form sodium acetate and water. The balanced chemical equation is CH3COOH plus NaOH make CH3COONA and H2O. This reaction occurs in a 1:1 molar ratio, meaning 1 mole of acetic acid reacts with 1 mole of sodium hydroxide. Therefore, By measuring the volume of NaOH required to reach the endpoint, we can determine the concentration of acetic acid in the sample accurately. On this slide, I will explain the titration apparatus used in this experiment. The main apparatus consists of a burette which is mounted vertically on the retort stand and secured with a clamp. The burette is filled with titrant, which in this experiment was sodium hydroxide solution. An aluminum flask containing the analyzed solution and a few drops of indicator is placed directly beneath the burette. During the titration, the sodium hydroxide is added slowly while the flask is gently swirled to ensure thorough mixing. The titration is continued until a permanent color change is observed, indicating that the endpoint has been reached. This setup allows accurate measurement of the volume of titrate used and help determine the concentration of the unknown solution. That will be all from me.
Hi, Assalamualaikum. My name is Nurul Shazlana Natasha and I will explain about filtration apparatus. So, filtration is a technique used to separate solid particles from a solid by passing the mixture through a filter medium. The main filtration apparatus includes a filter funnel which holds the filter paper and guards the liquid into a container. The filter paper acts as the filtering medium, allowing the liquid to pass through while retaining solid particles. A beaker is used to hold the mixture before the filtration, while an enlameyer flask or filter flask is used to collect the filtrate. For vacuum filtration, a puncture funnel and a vacuum pump are also used to speed up the filtration process. Besides, the purpose of filtration is to remove impurities from solution or to isolate any solid. There are two main types of filtration. The first type is gravity filtration. In this method, the liquid passes through the filter paper under the force of gravity. It is commonly used to remove impurities from a solution or to collect the filtrate. The second type is vacuum filtration. This method uses a vacuum pump to create suction, allowing the liquid to pass through the filtrate more quickly. It is mainly used when the solid residue is the desired product and needs to be collected efficiently. In conclusion, filtration is an important separation technique in analytical chemistry, and the choice between gravity filtration and vacuum filtration depends on whether the liquid or solid product is required. Okay, next is about heating apparatus. First, we have hot plate. Hot plate is electric heating device with a flat heated surface. Hot plates used to heat beakers, flasks and solutions safely. Second, heating apparatus is oven. Oven uses hot air to dry samples and glassware. Oven also maintain a constant temperature for drying. Then, third, we have furnace. Furnace produce very high temperature that is up to 100 degrees Celsius or higher. Furnace also used for etching, ignition and heat treatment of samples. Fourth is burner. Burner produce an open flame using gas. This also used for direct heating and sterilization. Fifth is water bath. Water bath heats samples indirectly using hot water. It is also suitable for gentle and uniform heating. Lastly, hot heating apparatus is desiccator. Desiccator is a tight container containing a desiccate. It is also used to cool hot samples and protect them from moisture before weighing. So this slide explains the importance of cleaning solution used in analytical chemistry laboratories. The main purpose of cleaning solution is to remove dirt, grease, stain and other contaminants from laboratory glassware. Proper cleaning is essential because any residue left on the glassware can affect the accuracy and reliability of experimental results. There are several common cleaning solutions used in the laboratory. First, detergent solutions are used to remove grease and general dirt. Second, we use dilute nitric acid. Dilute nitric acid is used to remove metal deposits and inorganic residues. Third, dichromate sulfuric acid solution is because it is effective for removing stubborn organic residues and stains. Then, we also use aqua regia, which consists of 3 parts of hydrochloric acid and 1 part is nitric acid. It is used for cleaning glassware contaminated with certain metal. Lastly, we use ultrasonic cleaning, are good for cleaning pipettes and burettes. It's because it can soak in warm detergent solution. Then, when handling this cleaning solution, safety precaution must always be followed. Laboratory personnel should wear gloves, safety goggles, and a lab coat. It because strong acid must be handled carefully, and aqua regia should only be used in fume hood because it releases hazardous fumes. After cleaning, all glassware should be thoroughly rinsed with distilled or deionized water to remove any remaining chemicals. In conclusion, proper cleaning of laboratory glassware helps prevent contamination, improve the accuracy of experimental results, and extend the lifespan of laboratory equipment.
In conclusion, analytical chemistry relies heavily on the proper use of apparatus, techniques, and reagents. Through the determination of acetic acid concentration in vinegar using acid-basic titration, we can see how analytical balance, volumetric glassware, titration technique, and chemical reagent work together to produce accurate and reliable results. Therefore, good laboratory practice are essential for successful chemical analysis.